Hi there, everyone. First, I'd like to acknowledge the Wurundjeri people as the traditional owners of the land I live and work on. I pay my respects to their elders, past and present, and any other Indigenous communities who may be listening. I'm going to be talking about environmental DNA, fire, and platypuses. To start off with, what is environmental DNA? Environmental DNA, or eDNA, is DNA that is sourced from an organism. So this could be anything from feces, skin cells, or even scales. Now, this comes from an environmental sample, which could be the air, soil, or in my case, water samples. Now, I'm using a single species detection method where we can go to a site and filter out some water. We can take these filters to the lab and extract DNA from them, and then use a species-specific primer which targets the DNA of just our target species, platypus, for example. We can then use qPCR to amplify the DNA and therefore determine the presence or absence of our target DNA in each of our samples. Moving on to the main questions of this presentation, I was interested in the effect of the 2019-2020 fire season on the occupancy of platypuses. There has been some studies looking at fire and platypus in the past, but these have some pretty variable outcomes. Platypuses could be impacted by fire because of phenomena such as sediment runoff, as well as the impact on their prey, such as invertebrates. Other studies have also been a bit hampered by a lack of systematic pre-fire data, as platypuses are so cryptic to see in the wild that this data can be hard to come by. However, we were sampling using eDNA for platypuses in 2018 and 2019 across southeastern Australia. And we've managed to resample after the fires 142 of those sites so far, which, considering the COVID restrictions in Victoria, was, I think, a pretty good effort. So therefore, we have samples from sites before and after the fires, as well as in impacted and control areas. We therefore decided to do a before-after control impact design. I'm going to be presenting results from the native vegetation areas, which is a bit of a reduced data set of 85 sites. So I'm using a hierarchical site occupancy detection model, which allows us to look at imperfect detection at the different levels of eDNA sampling. So first of all, we have our visits before or after the fire, and we have the probability of occurrence at each of these sites before and after the fires. We have the availability probability, which is the availability of DNA to be detected within each of our water samples. And we also have detection, which is, are we able to detect DNA using qPCR, given that it is available in our water samples and that platypuses occur at that site during one of our 2 visits. We've also included this extra information at the occupancy or occurrence level, such as whether the site is control impact, before or after, as well as the interaction between the 2. I'm just presenting the results we've got so far, which we have predicted occupancy, as well as before and after, as for the control and impact sites. Now, there was actually a difference in the predicted occupancy for the control and impact sites prior to the fires, which is an interesting result. But we have an increase in the predicted occupancy for the control sites and a decrease in the predicted occupancy of platypuses after the fires for the impact sites. So some general conclusions are that eDNA sampling was a pretty efficient way of sampling post-fires. The results so far suggest that there could potentially be a decline in platypus occupancy post-fires, but we're going to continue to investigate this. I'd like to thank my supervisors, collaborators, sources of funding, as well as acknowledge the traditional owners of the land on which this fieldwork took place.